Happy New Year, friends. Thank you so much for being patient. We're going to read our second part of Module 7, beginning on page 205. Section begins mitosis. Ready? Here we go. Mitosis, eukaryotic asexual reproduction. As you have already learned, the main component of a cell's DNA is stored in long, thin strands in the nucleus of the cell. If all of the DNA from one of your cells were strung together end to end, the string would dangle from your head to your toes, and still some would be on the floor. How does such a large amount of DNA get packed into a tiny cell? Examine the figure below. The DNA of a eukaryotic cell is tightly bound together with a network of proteins. Certain proteins, called histones, act as spools which wind up small stretches of DNA. The DNA wrapped around these histones form what could be described as beads on a string, which we call nucleosomes. Other proteins, including other histones, stabilize and support these spools, making a complex network of DNA coils and proteins. This network is called a chromosome. Chromosome, DNA coiled around and supported by proteins found in the nucleus of the cell. Most of the time, these chromosomes appear as long, fuzzy strands in the nucleus. As you learned in module number 6, we call this structure chromatin. When a cell is ready to reproduce, however, the coils form an even more compact version of the chromosome, which looks something like a dumbbell. This is often called a condensed chromosome, as it represents the smallest package in which nuclear DNA can be put. Now, as you might expect, it takes a lot of DNA to carry all of the genes necessary to code for the proteins of a living organism. Thus, there are several different chromosomes in the nucleus of the cell. What you might not expect, however, is that the number of chromosomes is dependent on the organism studied. For example, humans have 46 chromosomes in the nuclei of their cells, while a cat has only 38. Don't get the idea that more complex organisms have more chromosomes than less complex organisms, however. A horse has 64 chromosomes in the nuclei of its cells, and a crayfish, which you might call a crawdad, has 200. As we have already learned, there are two types of reproduction in God's creation: sexual and asexual. You have already studied the asexual reproduction of prokaryotic cells, but now we want to look at the asexual reproduction of eukaryotic cells. We call this process mitosis. Mitosis, a process of asexual reproduction in eukaryotic cells. Now remember, asexual reproduction results in offspring that have the exact same genetic code as that of the parent. Since this is the case, we often call mitosis cellular division, because in the end, the process makes two duplicate cells from a single cell. When a cell is busy with its normal life functions, it is said to be in a state of interphase. Interphase, the time interval between cellular reproduction. During this time, the cell is simply maintaining the status quo. The chromosomes are in the nucleus of the cell, but not in their condensed form. From time to time, sections of the DNA on the chromosomes unwind to begin the process of protein synthesis. Otherwise, the DNA remains in its chromatin form. The chromosomes are not as tightly packed as they could be, but they're still pretty tightly packed. The combination of interphase and mitosis is often called the cell cycle because the cell is either in one or the other. The cell spends most of its time in the interphase. When it is time for the cell to asexually reproduce, two things happen. First, the chromosomes must duplicate. After all, in order to make two cells, we need two sets of DNA. Thus, the chromosomes must make copies of themselves. Although this is a fascinating process, it goes beyond the scope of this course. If you are interested in learning about how this is done, visit the course website that we discussed in the student notes at the beginning of this text. It has links to websites that explain this fascinating process. After duplication, the chromosome and its duplicate are called sister chromatids. The second thing that happens to initiate mitosis is that the centrioles, which are outside the nucleus, duplicate themselves so that there are two sets of centrioles. Now that the DNA and the centrioles are duplicated, the cell is ready to begin mitosis. At this point, it is called a mother cell. Mother cell, a cell ready to begin reproduction containing duplicated DNA and centrioles. A mother cell is ready for prophase. In prophase, several things happen. The duplicated chromosomes coil into their condensed form, and they attach to each other at a point called the centromere. Centromere, the region that joins two sister chromatids. As you can see in figure 7.5, when the sister chromatids join together at the centromere, they form an X shape. This is the common representation of a chromosome. You have to understand, however, that chromosomes only look like this because they have been duplicated. The condensed form of an unduplicated chromosome is the dumbbell shape that we discussed earlier. Also during prophase, each set of centrioles migrates toward one end of the cell. Microtubules extend from the centrioles in all directions, forming the aster. As the centrioles migrate, microtubules from the aster grow, forming spindle fibers, which make up the mitotic spindle. As the mitotic spindle is forming and the centrioles are moving, the nuclear membrane breaks apart, exposing the chromosomes to the rest of the cell. Microtubules from each centriole attach themselves to the chromosomes and move them to the center of the nucleus. They line up the chromosomes along an imaginary line called the equatorial plane, which runs down the center of the cell, equidistant from the two centrioles. The brief time in which the chromosomes are lined up along the equatorial plane is called metaphase. At the conclusion of metaphase, the microtubules on the spindle pull the chromosomes toward the centrioles. Since each of the sister chromatids has a microtubule pulling on it from opposite centrioles, the sister chromatids separate from one another. One gets pulled to one side of the cell and the other gets pulled to the opposite side of the cell. This phase of mitosis is called anaphase. When the chromosomes are pulled far from one another, the last phase of mitosis, telophase, begins. During this time, the spindle begins to disintegrate and the plasma membrane begins to constrict along the equatorial plane, eventually forming two cells where there was once only one. A nuclear membrane begins to form around each group of chromosomes, and the chromosomes uncoil from their condensed form, forming the chromatin material of the nucleus. At this point, there are two daughter cells, where there was just one mother cell before. Please note that this phase is slightly different in plants, as we'll see in a moment. The process of mitosis is rather long and complicated, so it certainly deserves some review. Examine the figure below, which summarizes the process of mitosis. Now, you might wonder what happened to all of the other organelles in the cell during mitosis. Well, they are all still present while mitosis is occurring. We simply chose to ignore them in our discussion because we wanted to concentrate on just those organelles that are important to mitosis. It turns out that the division of organelles between the two daughter cells is regulated during the process of mitosis. Each daughter cell gets at least one of each organelle. If there was only one of a given organelle prior to mitosis, it is divided between the two daughters. But wait, how does each daughter get along with only half of the organelles or with only part of an organelle? Well, remember that the DNA in each of the cells has all of the information necessary to construct any organelles that the cell needs. Thus, the cells can build more of each organelle, and they can also complete any partial organelles that they have. In fact, since mitochondria have their own DNA, they can actually replicate themselves. As a result, if there is only one mitochondrion in a cell during interphase, it can actually replicate during mitosis so that each daughter gets one. Figure 7.6 might have been easy to follow since it was an idealized schematic drawing. In real life, of course, things are a little more difficult to see. Examine the figure below to see what mitosis looks like in animal cells when they are viewed under a microscope. When you think of asexual reproduction, you probably think of organisms like bacteria, protozoa, and perhaps fungi. Most of the organisms with which you are familiar, dogs, cats, humans, etc., however, reproduce sexually. This means mitosis is irrelevant in the study of these organisms, right? Wrong! It turns out that nearly every eukaryotic organism uses mitosis. After all, when you are born, you are tiny. As time goes on, you grow. How does this happen? Well, your body produces more cells. How are those cells produced? By mitosis. Also, even after you are fully grown, your cells must continually go through mitosis in order to replace cells that have died. For example, millions of red blood cells die every minute in your body. If they weren't replaced, you would soon run out of them. How are they replaced? By mitosis, of course. So you see that mitosis is relevant to nearly every creature in God's creation. Now, the mitosis you just learned about occurs in animals. A slightly different kind of mitosis occurs in plant cells. The major difference between plant and animal mitosis is found in telophase. Because of the rigid cell wall in a plant cell, the plasma membrane cannot just constrict as we learned in animal cell mitosis. Instead, once the chromosomes have been pulled to the centrioles, vesicles containing the building blocks for the plasma membrane line up along the equatorial plane. These vesicles begin fusing together, forming a new plasma membrane. As the new plasma membrane forms, cellulose is formed in the middle, forming a new cell wall that splits the plasma membrane in two. Eventually, even a middle lamella is formed, resulting in two cell walls, two plasma membranes, and thus two cells. If you have a microscope, you can get more experience identifying the stages of mitosis by performing the following experiment. If you do not have a microscope, you can skip the experiment. Diploid and haploid cells. As we mentioned before, humans have 46 chromosomes. What we fail to mention, however, is that these chromosomes come in pairs. Thus, in a human cell, there are 23 chromosome pairs for a total of 46 chromosomes. The two members of a chromosome pair are very similar, but they are not identical. We will come back to this point in a little while. Because the members of a pair are similar but not identical, the pairs are called homologous chromosome pairs. The term homologous means similar but not identical. Each member of a homologous chromosome pair is called, reasonably enough, a homologue. Now, this can get a little confusing, so we will try to illustrate it with a figure. Suppose we were to take the chromosomes from a human cell during the metaphase of mitosis. Remember, at that point, the chromosomes are attached to their copies at their centromeres. This gives them an X shape. If we were to look at the chromosomes at that point, we would get something that looks like the figure below. Notice that the chromosomes are grouped in pairs. The two chromosomes in each pair look incredibly similar, but this is not because the chromosomes have been duplicated for reproduction. The X shape of each chromosome is the result of the fact that the chromosome has been duplicated because each chromosome is attached to its duplicate, its sister chromatid, at the centromere. The fact that the chromosomes come in pairs is simply a property of the nuclear DNA in a human cell. Thus, each cell has two of chromosome number 1, two of chromosome number 2, and so on. The only exception to this is found in males. As shown in the figure, if a cell has similar chromosomes in its 23rd pair, the cell must be from a female. If not, it must be from a male. Since this pair of chromosomes can be used to distinguish between the sexes, they are called sex chromosomes. By convention, the two sex chromosomes in a woman's cell are called X chromosomes, while the chromosome that distinguishes a male cell is called the Y chromosome. As a result, we say that females have two X chromosomes, while males have an X and a Y chromosome. We will learn a great deal more about that in the next module. As a point of terminology, a figure like figure 7.8 is called a karyotype. Karyotype, the figure produced when the chromosomes of a species during metaphase are arranged according to their homologous pairs. In this particular karyotype, you can see that there are 23 pairs of chromosomes, chromosomes 1 through 22, and a pair of sex chromosomes. With the exception of the XY pair in males, each homologue within a pair has exactly the same number of genes as its partner. These genes also code for the same trait. For example, you will learn in the next module that whether your blood type is A, B, AB, or O depends on a specific gene. That gene is located on the chromosomes of pair number 9. Each homologue in the pair has a gene for blood type, and you can find that gene in exactly the same spot on each chromosome. Now remember, these chromosomes are not identical. They are similar. Thus, they each have a gene for blood type, and that gene is found at the same place on each chromosome. However, one gene might be for type A blood, and the other might be for type O. In the next module, we will learn the implications of this fact. When a cell's chromosomes come in pairs, we say that it is a diploid cell. Diploid cell, a cell with chromosomes that come in homologous pairs. Many species have diploid cells, and this results in a great deal of diversity within the species. Cells with chromosomes that do not come in homologous pairs are called haploid cells. Haploid cell, a cell that has only one representative of each chromosome pair. As we will see in the next section, even species that have diploid cells will have some haploid cells. Before we leave this section, we need to make sure you understand what we mean when we talk about the number of chromosomes in a cell. When we say that a horse has 64 chromosomes, this means that it has 32 pairs of chromosomes. The total count is 64, but they come in 32 pairs. Since this terminology can be a little confusing, we sometimes make it a little clearer by referring to the diploid number, sometimes abbreviated as 2N, or the haploid number, sometimes abbreviated as N. Diploid number 2N, the total number of chromosomes in a diploid cell. Haploid number N, the number of homologous pairs in a diploid cell. Based on these definitions, we could say that the diploid number, 2n, of a human cell is 46, while the haploid number, n, is 23. Now, remember, a haploid cell has only one chromosome from each pair. Thus, the haploid number is also the number of chromosomes in a haploid cell. Well, friends, that will end part 2. We'll pick up in just a little bit and go on talking about meiosis. See you soon.